Yeah, right. My name's Mrs. Chiverton. I'm the Science Director, and I'm going to be taking you through Lesson 5 in the Year 8 series about hormones and auxins. So before we start, as always, can you remember to turn off any notifications on any devices you are using to watch this video? And make sure you have either got a pen and a piece of paper, or better still, your exercise book, ready to record and do the activities that are part of this lesson. There is an optional practical that you may wish to do, but it doesn't stop you taking part in the lesson if you cannot do it. So what you will need are some fast growing seeds such as cress or mustard. You can often buy these in supermarkets or obviously garden centres, places like that. 3 saucers of similar shallow dishes. They don't need to be particularly big. Some kitchen roll or some cotton wool, either one works just as well, and permission to leave these things sitting around on a windowsill or somewhere similar for at least a week. If you need to go and collect any of those things, just pause the video now and then you'll be able to continue with it as we move along. So as I've said, today's lesson is part of the Hormones and Auxins series. It's lesson 5 and it's all about plant hormones and growth. So our starter is a recall from stuff that you should have covered at Key Stage 2. You've got a diagram with the basic organs of a plant there, A, B, C and D, and I've given you the labels to match to them: stem, roots, leaf and flower. So you should be able to write down the letters and match them to the correct name of that particular organ. And then for many of you, I'm hoping you will challenge yourself and do the extension activity to remind us what each of those parts of a plant actually does. And I'm going to give you a minute and a half to complete that activity. Off you go. Okay, hopefully you've managed to get that done. If not, just pause the video and you'll be able to carry on with that. But let's look at our answers. So we should have had part A being the flower, and that's involved in reproduction. Part B is the leaf, and the leaf is involved in photosynthesis to make food for growth. Part C is the stem, and the stem is for transport of substances to and from the leaves and the roots. And part D would have been the root, and that has the purpose of absorbing water and minerals for growth, and of course it also anchors the plant into the soil and stops it blowing away. So hopefully those are all correct, and if not, you can just remind yourself about those for the future. So we're in part of our learning journey on homeostasis and hormones and auxins, and we've looked at various things to do with humans, and now we're moving on to. So in today's lesson, I'm hoping you are going to be able to investigate the growth of roots and shoots, and also to be able to plan a valid investigation. And that word valid is one of our keywords that we're going to look at in quite a lot of detail. So What I want you to do in your exercise book is to list the 5 things that plants need to grow. And I've given you 5 pictures there that should give you some clues as to what 5 things are needed for successful plant growth. I'm going to give you a minute to look at those pictures and try and write down the 5 things that plants need in order to grow. Okay, so you should have a good idea of some of these, at least looking at those pictures. So hopefully we'll have worked out that they need sunlight, they need a warm temperature, they need minerals that come from the soil, they need water, which again is absorbed from the soil through the roots, and they need carbon dioxide that they take in through their leaves. And that's in order to do photosynthesis and then to make the other complex chemicals that the plants need for growth. And if any one of those is limited, then that is going to cause problems for plants growing. And you'll look at that in more detail in other modules and certainly at GCSE. But photosynthesis can't happen until the plant has actually started growing and developing leaves. So most plants actually start their lives as seeds, and those seeds have stored inside them all the nutrients that the plant needs for that initial growth period. They're usually scattered away from the parent plant, and that can happen by the wind. So think about dandelions, for example, with their little parachutes flying around, or by animals. So anything that is a seed or a fruit that would be usually transported by animals. And that's so that they don't compete with the parent for the resources that they will need to grow. So if we think about all the variety of seeds that there actually are, when those seeds land in the favourable conditions, they will start to germinate. And what seed germination is the process by which a plant grows from a seed. It's a sealed storage unit, but eventually it does have to grow. And you can see at the bottom there is a whole set of stages where we get initially an embryo plant and the seed coating eventually breaks off. We get something called cotyledons. That's very, very immature leaves, and that can start the photosynthesis process. Meanwhile, the roots are growing underground to get the water and the minerals that are needed. And eventually a shoot will fully develop with actual leaves and photosynthesis will happen. But in order to get to that stage, the seeds need certain conditions to grow successfully. And that is oxygen, it's water, and again, a particularly warm temperature in most cases. So what I'd like you to do now is to record in your exercise book or on your piece of paper what germination actually is and what are the 3 conditions that we need for germination. And I'm going to give you 1 minute to get those down. It will only be a couple of sentences to complete that answer. So just checking and amend any of your answers if you've made any mistakes, but germination is the process by which plant grows from a seed. And seeds need oxygen, water and warmth to germinate successfully. So what we're going to do is we are going to investigate how seed germination occurs. And when we think about investigating things, what could we actually test? How could we actually measure this? So if we are talking about seed germination, we would need an independent variable to investigate. That's the thing that you could change. And so it's got to be something that would affect seed germination. So the obvious things are to think about what a plant needs for seed germination. Then what would the dependent variable be? Now, the dependent variable is the thing that we measure that changes during our experiment. So we would have to be something to do with germination, either the amount of seeds germinating or the amount of growth that's happening. And then to make our investigation valid, of course, we need to control any other variables that could affect the germination rate. So anything else we would need to keep the same, and that's what we would need to control. So have a think. We know that germination needs oxygen, it needs water, and it needs warmth. So which one of those, are there any of those that we could have as our independent variable? If once you've decided on that, what would our dependent variable be? How are we going to measure germination? And then what else would we need to keep the same in order to be sure that we've got a valid experiment? So write down any answers to those questions, please. And I'll give you 1 minute just to record your answers. So our independent variable could either be temperature, water or oxygen. Any one of those three is a suitable answer. The dependent variable is likely to be either how many seeds actually germinate or how big the seeds grow in a set period of time. And then our controlled variables would have to be the type of seeds that we use, and whichever other variables is not your independent one. So if you chose temperature as your independent one, we would have to control the amount of water and the amount of oxygen. If you chose water as your independent variable, we would have to keep the temperature the same or the amount of oxygen the same. And if we chose oxygen as our variable, we would have to keep the water and the temperature the same. So we're going to look at investigating temperature because that's the easiest one that you might be able to carry out at home and we would be able to carry out in school. So if we investigate the temperature, that's going to be our independent variable, and we're going to measure how many seeds germinate in a period of a week. So that's going to be our dependent variable. So what I want you to do now is think about how we will make our results valid and what that word valid actually means. So have a go at writing that down in your book. What do we mean, or how will we make the results valid, if that's what we're going to investigate, and what does that word valid actually mean? 1 minute. Okay, so validity is how well a scientific test or piece of research actually measures what it's set out to do. In other words, how true the results actually are. And by controlling other variables, that's what makes our test valid. So we would need to be controlling things like the volume of the water and the number of the seeds that we are doing. And validity can be improved with accurate measuring and producing reproducible results. So this is what will be done in the lab, but we can adapt it for a home environment. So in the lab, we would take 3 Petri dishes and we would line them with cotton wool. We would measure out 10 mils of water, so that's one of our controlled variables, and we would pour it evenly over the cotton wool. And we would repeat step 2 for the other 3 dishes. We would then count out 10 seeds for each dish. That's our second controlled variable. And again, we would place the seeds spread evenly in each dish. And again, that's controlling it because we're not bunching them up together, we are spreading them evenly around. We would label the dishes with temperatures 0, maybe 18 degrees and 50 degrees C. And each dish would be placed in the appropriate temperatures and the results will be checked next lesson. So 0 degrees, we would put them in a fridge. 18 degrees would probably be room temperature, and 50 degrees we would have to put them in an incubator to keep them at that temperature. Now I know you won't have all of that equipment at home, but what we could do is we could use saucers, so 3 saucers or similar dishes. It could be lids of things, as long as they'll sit flat, and we can line them with kitchen roll, or if you've got cotton wool at home, you could use cotton wool. If you measure out 1 tablespoon of water and pour that evenly over the kitchen roll, that will control the amount of water, and do that for all 3 dishes. We still need 10 seeds for each dish, but if you haven't got 30 seeds altogether, just divide your seeds evenly between the 3 dishes and spread them evenly across. Now, we're going to label them cold, room and warm. And what I would suggest you do is put 1 in the fridge, that'll be our cold one. 1 in a room, somewhere away from direct sunlight, so just you know, in a bedroom or something, and then 1 on either a very sunny windowsill or close to a radiator, and that will be our warm one. And we can check these in about a week. So if you are going to do this experiment, I would suggest you pause the video now so you can see the method, and you collect your equipment and you start and you set that up. It will only take about 5 minutes to set it up. Once you've done that, you can restart the video and we'll look at what we would do with some results and about validity. So one of the things we often do when we do experiments is we come up with a hypothesis, and a hypothesis is something that we can test, and we would make a prediction. So in your exercise book or on your paper, put the title Plant Hormones Investigation. And what I want you to do is write a prediction for the experiment and include in that a scientific reason for your answer. So out of your 3 dishes, which one do you think will germinate the best, and what is your reason why? Or alternatively, you could look at it the other way around. Which one do you think would germinate the least, and your reasoning why? Or you could actually predict a pattern. So the hotter the area, the lower the germination. This happens because. So what we're doing is making a prediction, saying what we think will happen, and then trying to give a scientific reason for your answer. And remember, this is all to do with things that can affect germination. So I'm going to give you 2 minutes to get a prediction and a scientific reason recorded in your books. So just make sure we've used our proud and we've checked our spelling, our punctuation and grammar. Have we made a prediction of which temperature will have most seeds germinated? And have you given a scientific reason why that includes the requirements for germination? I'm just going to give you 1 more minute to look at that again and just amend anything that you need to make sure you fulfill the criteria. So we've made our predictions and we'll leave our seeds to germinate and then we'll look at the results of them next week. So another group decided to investigate the effect of pollution on seed germination, and here are the results that they have produced. So their independent variable was the concentration of polluted water, and they went from pure water as a control, so they had something to compare it to, and then they had different percentages of pollution. They used 50 seeds every time, so that was one of their controlled variables, and they counted how many seeds of those 50 had germinated at the end of a set period of time, which is another control variable, and then they worked that out as the percentage of germination. And we quite often get results like this from our experiments. And then we have to process that data and see if there are any patterns in our data and if they match the predictions that we made in the first place and what scientific reasons there can be behind those patterns. So what I'd like you to do is look at this data and see if there are any conclusions that you can draw from these results. And you really should use evidence in your answers. So pick out numbers to compare. How could the data be better presented? So we've presented it in a table here, but is there something else we could have done with it that might have made it easier to see a pattern? And if these results were from a real experiment, how could a gardener or a farmer use these kind of results to their benefit? So I'm going to give you 2 minutes to have a think about and have an answer to those questions written in your exercise books. Off you go. Okay, so what can we conclude from these results? Well, there is a pattern there to say the higher the concentration of pollution, the poorer the germination rate. You could also pick specific data out of there and say that it got worse the higher the level of pollution, the lower the germination rate. And you could pick out the points where no seeds were able to germinate. So lots of different conclusions that we could come from that. How could the data be better presented? Well, if we drew that as a graph, it would be a lot easier to spot a trend or a pattern. Because the independent variable didn't increase at an even rate, it was quite tricky to know when the changes happened. So that would allow us to see that more clearly. And how could a gardener or a farmer use these results? Well, what they could do, they could test their water and find out what the levels of pollution are, and then they could use data on seeds to find out what will grow best in that water, because some seeds will tolerate higher levels of pollution than others, and they can use those ones to make sure that they're going to get the best possible crop. So these kind of investigations do have a real life application where farmers and growers can actually use that information to end up with the best possible crop and therefore the best profits for them. So this is a little bit of a look forward to GCSE. If you watch the two little videos here, what are those plants actually responding to? Because we don't think of plants as moving, but you can see very clearly here that actually those plants are moving. Now what's happening is those plants are actually growing towards light, and you can actually see that happening over a 24-hour period, that seedlings like this will move around to track the light if they're in a sunny window. And the one on the right hand side there, you can actually physically see the leaflets closing up in response to being touched by the person. Now, both of these do this because of a set of chemicals called auxins, and these are plant hormones. So we've talked a lot about human hormones, but these are plant hormones. And you will do a lot about auxins as part of your GCSE science. But please be aware that plants do actually move and respond to things like light and touch, water, and even gravity. So, if you've been given a Google quiz to accompany this lesson, I suggest you do that now, and it will be automatically marked for you. And here are some additional notes just to make sure you have all of the key points written down to add to your notes. So germination is the process by which a plant grows from a seed. Seeds need certain conditions to grow successfully, so that's oxygen, water and warmth. An independent variable is what we choose to change in an experiment. The dependent variable is what we measure during the experiment, the outcome of our experiment. And controlled variables are what we keep the same in order to ensure an investigation is valid. Thank you for listening today, and I'll see you in one more hormones lesson where we'll look at the results of our experiment.